Welcome. Welcome to this session of pharmaceutical analysis. Today, we're going to understand that what are the specific quantities which are used in the pharmaceutical analysis and the concentration. This is how we can express the concentration. This is how we measure the concentration. So now let's move to the pharmaceutical analysis. As far we have understand about the pharmaceutical analysis is that this analysis it is used for the purpose of identification, quantification, right? And this identification is a part of the quality. And as we have already discussed that the analysis can be qualitative analysis and the quantitative analysis. So we can say that analysis it is intended to either identify or quantify one or more than one substance in a given sample. What is a sample basically? Sample which consists of one or several analytes. What is analytes? Analyte is a substance which is to be identified or quantified. So when these analytes of one or more than one analyte, they form the sample. A sample matrix is also the rest of the sample. It is there in the sample, but this is the rest of the sample than the analyte. This is known as sample matrix. Now the identification, what is identification? Is to confirm the identity of the analyte. This identification also be referred as a qualitative analysis. One of the parameters of the qualitative analysis. What is quantitative analysis? Quantitative analysis is used to measure the exact concentration or amount of the analyte in a given sample. So when we are doing the quantitative analysis to measure the exact concentration or the amount in a given sample, then we have to some unit to express the concentration. We need some method or some expression for the concentration. Now while we are starting working on our sample, we got a sample, we receive a sample. Now we store this sample, or maybe this is a compound which we buy from we buy from the market, and we stored in a sample storage room. Now in the sampling, what is the sampling basically? So this sampling is a very broad term which is different meaning for the different fields. But in the pharmaceutical analysis, this sampling represents the sample. This sampling represents the preparation. It is a it involves the preparation. It involves the storage. It involves the analysis or the measurement. The sample storage is also very important, and the reason behind it is to avoid the compositional changes of the sample during the storage. So to protect the sample, it should be stored at very low temperature, protected from light, refrigerator, or some stored also in freezer. So after the sampling and storage, when samples are normally pre-treated in some way or the other way, and this is known as, or this is called the sample preparation, which is a pre-treatment of the sample. So this sample preparation can be very simple or sometimes very complicated depending on the sample. Thereafter, after the sample preparation, first thing is why we need sample preparation. So we need the sample preparation for so that the sample can be analyzed in a very subsequent steps. The sample is compatible with the final analytical method or instrument. The analyte which is present in a sufficient amount is to be detected. Substance in the sample matrix that can cause problem or interference are also removed. Now, after ensuring the sample preparation, the analytical measurement is performed. And this measurement where the analyte or analytes they are identified and quantified. So now the question is how to specify the quantities and concentration. So in most of the cases, a quantitative pharmaceutical analysis is performed when the analyte is present in a solution. What is this solution? A solution is a homogeneous mixture of two or more substances. Now, a minor species in a solution is called a solute, and the analyte is an example of the solute. The major species in a solution is called the solvent, and the amount of analyte, that is the solute, is normally expressed as the concentration. This concentration means that the amount of the solute per volume unit of a solution. What is it? Amount of solute per volume unit of a solution. So now what is molarity? This is one of the way to express the concentration. So in some context, the term molarity is used to express the concentration, which is abbreviated as M, and you can see that the molarity of a certain solute is defined as number of moles of solute per liter of solution. So what is molarity? Moles of solute divided by liter of solution. That means number of moles of solute per liter of solution is known as molarity. Now when the concentration are low, it is impractical to use the molarity M. In such cases, we use millimolar, micromolar and nanomolar. So just like it can be seen that 1 molar is equal to 10 to the power 3 micromolar. Now, which is equivalent to 10 to the power 6 micron molar, which is equivalent to the 10 to the power 9, that is nanomolar. There is an alternative to the molarity, and it is very common to use the concentration expressed as mass per volume unit. Expressed as mass per volume unit. Either milligram per ml, which is mg per ml, microgram per ml, μgram per ml, nanogram per milliliter, ng per ml, or also sometimes picogram per ml. It can also be used. Now let's understand the molarity. How can we calculate the molarity? By few examples. So see, this is one of the example. We have 0.100 gram paracetamol, which is dissolved in water, and the total volume is adjusted to 500 ml. So in 0.100 gram of the paracetamol is dissolved in water, and the volume is made up or adjusted to 500 ml. How can we find out the molarity? So we know that the molarity is number of moles per liter of solution. So number of moles of solute. So here is the paracetamol is the solute. What is the number of moles? This is the molar mass. What is the molecular weight of the paracetamol? If we calculate by the formula, so then we found that the molar mass is 151.2 gram per mole. It is the molecular weight or the chemical weight of the paracetamol. also known as acetaminophen. So now this 151.2, it is the gram per mole is the molar mass of the paracetamol. So we take 0.100 and dissolve it in water and make up the volume up to 500. So now we have to find out that how many moles are there in 0.11 gram. The molar mass is 151.2. So now calculate the number of moles. So weight divided by molar mass, right? And this is number of moles. When the number of moles comes, it is further divided by the volume, that is per liter. So making the 500 ml first into liter divided by, how can we change the liter, ml to liter, by divide by 1000. So it is 0.500. So 0.100 gram divided by 151.2 gram, it is number of moles in 100 gram. It will calculate the number of moles in 100 gram, and which is further divided by the volume, which is in liter. So then the molarity will be 1.32 into 10 to the power 3 molar. Then we can also convert it to millimolar or micromolar. So just like see in the example that one sample has a content of 1.62 into 10 to the power minus 5 molar of paracetamol. How can we convert into micromolar? So as we know that 1 molar is equal to 10 to the power 6 micromolar. So 1.62 into 10 to the power 5 molar multiplied by 10 to the power 6 micromolar. That gives the micron 16.2 micromolar. Now one more example, that now a sample is given to us the content of 12.5 milligram per ml. That is the concentration already given, right? In the earlier cases on the above, only the weight of the paracetamol was given, which is 0.100. Now here in this example, 12.5 milligram per ml of the paracetamol is there. We know that molar mass of the paracetamol is 151.2 gram per mole. Now how can we convert this milligram per ml to the molar? So first 12.5 milligram per ml, convert it into gram per liter, right? Because number of moles is in grams per mole, right? So that's why we have to convert milligram into gram and ml into liter. So for that 12.5 multiply by 1000 and also divide by 1000, because milligram to ml, milligram to gram and ml to liter. So that will 1000 cut by 1000, nullify by 1000. So again the concentration would be same, that is 12.5 gram per liter. Now how can we calculate the molarity? First of all, in this 12.5 gram per liter, because the liter is already there, so just we need to convert it into the number of moles. So 12.5 gram per liter divided by 151.2 gram per mole, that is, then the molarity would be 8.27 into 10 to the power minus 2 molar. So this is how we calculate the molarity. We have a number of experiments in our practical classes, so we will perform such kind of conversions in our practical classes too. Now moving to the next slide, that is how we have also some other option. We have other option for the concentration, that is concentration percentage. So concentration can also be expressed as a percentage. You can see that there are following definitions which are used, like percentage weight by weight, percentage volume by volume, percentage weight by volume. So when we express the concentration as a percentage, it is very important to specify that whether the percentage weight or percentage volume or percentage weight by volume has been used. Now the percentage weight is used to express the concentration of solid or liquid, either in solid sample or liquid sample, right? Percentage of the weight is used to express the concentration of solids or liquid either in solid sample or liquid samples. Percentage of volume, however, it is used to express the concentration of liquid in liquid samples only, or gas in gaseous sample, that is volume. Percentage weight is used to express the concentration of solid or liquid either in solid samples or liquid samples, while the percentage volume is used to express the concentration of liquid in liquid sample or gas in gas samples. Now the third one is the percentage weight by volume. It is used to specify the concentration of the solid or liquid in a solution. Solid or liquid in a solution. So weight is weight in weight or volume in volume, or percentage volume in only to express the concentration of liquid in liquid sample or gas in gas sample, and the percentage weight by volume is used to specify the concentration of solid or liquid in a solution. Now see, this is an example that 5.01 gram of paracetamol is dissolved in 200 ml of ethanol. This ethanol is an alcohol. So, and the total weight of solution is determined by 162.81 gram. The concentration of paracetamol in percentage weight is, we have to calculate the weight by weight. So now the weight here is 5 gram. Now as we are calculating it percentage weight, so weight in weight, so it is divided by the total weight, which is 162.81, and then multiply by 100, that gives 3.08% weight by weight. That the 3.08% of the 162, it is 5.01 of the paracetamol, which is dissolved in 200 ml of the ethanol. This is percentage, concentration percentage. Now moving to the next is the ppm and ppb. This ppm and ppb is used when we have a very low concentration of sample. This is ppm is parts per million and ppb is parts per billion. So when we have a very low concentration, it is, or it may be convenient to use the term parts per million or parts per billion instead of the percentage weight. So parts per million, we can see, this is the formula: mass of the solute divided by mass of the total sample into 10 raised to the power 6. ppb, mass of the solute, because the million is 10 to the power 6, so that's why it is multiplied by 10 to the power 6. Parts per billion, mass of the solute divided by mass of the sample, and it is multiplied by 10 to the power 9. So actually, for the analysis of the pharmaceutical raw material and the preparation, the analyte concentration must be relatively high, and it should be normally milligram per ml to express the concentration. However, when we are analyzing a drug in the biological samples, the analyte concentration, they are normally very low. So in those cases where there is a very low concentration of the analyte, then we use the nanogram. We can also use the nanogram per ml or molarity, but we use generally the parts per million or parts per billion. Let's see, this is an example to convert the gram into the parts per million. We have 0.106 gram of ethanol. However, the ethanol is in liquid, but we can convert it to gram by the density is equal to mass by volume. So it is dissolved in 999.911 gram of water, the concentration of ethanol in parts per million. So for that, we can take the weight of the ethanol plus the weight of the total sample, that is as per the formula, and then multiply by 10 to the power 6, and that gives to 106 parts per million. So this is the molarity, percentage concentration, parts per million, parts per billion. These are some of the expression to show or to measure the concentration. Next time we will do some more kind of the measurement or expression of the concentration. Till then, thank you.